Welcome to RotaChrom's latest webinar. I'm Ben Solovas, and joining me in today's insightful conversation is a distinguished expert in the field, Arpad Konczol, who is also the lead scientific officer of RotaChrom Technologies. Arpad, it's an honor to have you here.
Hi, Ben. Thank you for this nice introduction. I'm really happy to share our brand new results from the RotaChrom Research Lab in this webinar.
Thank you so much. We're looking forward to hearing all of that. And speaking of that, let's talk about the agenda. In this episode, Arpad is going to talk about three things. First, the latest findings, as you just mentioned, from an extensive feasibility study. Then tackling a challenging separation task with significant implications for the pharmaceutical sector, conducted in close collaboration with esteemed academic groups. And finally, focusing on the advanced operation modes of the CPC. So it's going to be a very exciting webinar. And Arpad, I'm going to let you speak now. Feel free to take away from here.
Thank you, Ben. So ladies and gentlemen, thank you again for joining us today in this webinar. The subject of this today's talk is the preparative separation of Remdesivir diastereomeres by liquid-liquid chromatography. And yes, it was an extensive work with a lot of learnings. In the first part of my talk, I'm going to introduce the basic working principles of centrifugal partition chromatography. Then I will present the target compound of this study, the Remdesivir diastereomeres, and the unmet need that we discovered during this study related to Remdesivir. In the second part, I will detail the solvent system screening in this study, so the quest for the gaining selectivity in this scenario, and the mental traps that we faced during this work. I guess the most important part of my presentation is coming as the fourth point of this talk, is the resolution enhancement in CPC and different advanced, so-called advanced operation modes of liquid-liquid chromatography. And in the conclusion, I will shortly discuss the diastereomer separation in general, so just touching the chiral or non-chiral methodologies that can be used in such separation task. So the basics of CPC. In CPC, the separation occurs between two immiscible liquid phases. So both the mobile and the stationary phases are liquids. In our case, the chromatographic column in CPC is a rotor. So as you can see on the left-hand side, these are so-called extraction cells interconnected in series by ducts. And we use a strong centrifugal force to immobilize this stationary phase inside the rotor. Here you can see this on this scheme that CPC can be operated in two ways. So both the upper phase and the lower phase can be used as mobile and stationary phases. So the main point here is that the role of the phases is interchangeable, and we can use the system in so-called ascending mode. And as in the top chart, you can see that the mobile phase is moving against the centrifugal force in a bottom-up direction, and it can be interchanged, and the more dense lower phase can be also utilized as the mobile phase. In this case, the setups looks like in this way. So yes, the separation mechanism in CPC is really simple. It's just based on the different partition behavior of the components in the mixture. And so the method development is also really easy and straightforward. We have to focus on to maximize the solubility of the target input in the solvent system, generate selectivity, generate high stationary phase retention, in the rotor, in the CPC system, and optimize the partition value of the target compound in this so-called sweet spot range. And so, as you can see on this chart, once we measure the partition coefficient of the components in the so-called shake flask assay, then practically we can predict the chromatogram, and this is also true vice versa. So, coming to the target compound of this study, here you can see, and I guess unfortunately most of the audience is familiar with this compound. This is a blockbuster drug called Remdesivir. This is a broad-spectrum RNA polymerase inhibitor, and it contains a chiral phosphorus atom. It is authorized to treat COVID, several COVID symptoms. And what is the most interesting feature of this compound, that initially a chiral HPLC was utilized to separate the two phosphorus diastereomers of Remdesivir. And yes, it is formulated with sulfobutylated beta cyclodextrin in an infusion to get solubility, proper solubility for Remdesivir. And what is also important to mention is that the RP isomer has also clinical significance of the compound, but the SP isomer is the authorized compound. So the goal of our study when we started this story was to develop different centrifugal partition chromatographic methodologies for the efficient and scalable separation of these phosphorus diastereomers of Remdesivir. And how we started the study, as I mentioned, cyclodextrin is used in the formulation of Remdesivir. So we have had a preconcept to use this cyclodextrin-based solvent system, since cyclodextrin showed resolution in capillary electrophoresis in HPLC. It provides a nice stability constant and a nice improvement in the aqueous solubility of the target compound. So we performed a huge solvent system screening campaign, and what we found is that the partition showed a trend with pH, cyclodextrin concentration, and temperature. So that values, that results was what we anticipated, but unfortunately the selectivity showed no trend with these variables. And also the most interesting or most well-performing solvent system in shake flask methodologies showed very moderate or very low selectivity. So at that point we were considering to terminate the study, but luckily we discovered that the non-chiral resolution in reversed phase HPLC is also really nice for these diastereomers, and we decided to go with non-chiral solvent systems, and we concluded that no chiral recognition can be utilized in liquid-liquid chromatographic systems with using cyclodextrins. So as a next step, we screened more than 50 solvents to get the proper partition solubility and selectivity for Remdesivir, and as you can see, Remdesivir is not soluble in water, but it's also not soluble in alkanes and aromatics, and also in ethers. So this is a really tricky solubility profile, so to say. But we found that different polar co-solvents that can used as a really nice co-solvent in the different solvent systems, such as methanol, dimethylformamide, dimethyl sulfoxide, methyl THF, and some exotic ones. But the main message is here that water-free, so non-aqueous systems cannot be utilized. So here you can see in this large table the best solvent systems that we have identified, such as different alkane, co-solvent water systems. We came up with this solvent system concept, with this quaternary solvent systems concept, and we identified methyl THF with different alkanes as the best one to generate selectivity for the diastereomeres in non-chiral environment. So here you can see that we managed to increase the selectivity significantly, but in this case we were able to increase selectivity up to 1.5, which is a really high value considering, for example, preparative HPLC. But in liquid-liquid chromatography, this is the so-called threshold for baseline resolution. And on the next chart, I will detail the background for this 1.5 selectivity value. Here you can see the resolution of the two peaks in the function of the partition coefficient of the first peak, and the blue colored curve shows a typical classic elution case in liquid-liquid chromatography with a selectivity value 1.5 and the theoretical plate number 600. And as you can see, the desired 1.5 resolution value can be easily achieved in this scenario. But in our case, with lower selectivity values such as 1.3, as represented by the red curve, the 1.5 resolution value cannot be realized in this case. So this is can be considered as an insufficient selectivity. Okay, but what can be done in such cases in liquid-liquid chromatography? Since we have, as I mentioned in the introduction, since we have just liquids in our system, so both the stationary and the mobile phase, as these are liquids, we have the option to use the so-called dual mode. It means to reverse the role and the direction of pumping of the phases. So just switch between the phases and pump back the unresolved peaks after one column volume and get higher resolution values. Here you can see on the yellow curves that after one and after two column volume, if we reverse the phases and increasing virtually the plate number of the separation, we can get the desired 1.5 resolution in such way. And just for your reference, I also included in this chart the green curve representing 1.3 selectivity values with a 2.5 times higher theoretical plate number. So that means that if you consider this point, this working point, that by reversing 2 times the compounds in the CPC system, we can get 2.5 times higher theoretical plate number. So this is the way how we can increase the column length in CPC. And yes, there are a lot of options, such as the recycling mode, the so-called multiple dual mode. I will share more details on that approach. And most interestingly and the most efficient approach is the so-called dual rotor approaches, which I will also explain in more details on the next slides. Okay, but what is this MDM, or so-called multiple dual mode? Here you can see a typical setup of a CPC system with just 5 instruction cells representing as the rotor and binary input material, compound E and compound B. And here you can see that compound E has a lower partition coefficient, and compound B has a higher partition coefficient. And if we fill up the rotor 50-50 with the upper and the lower phase, and just eluting, injecting the sample with this A plus B, then the sample distributes between the cells, and compound E, since it has a lower KD value, travels faster in the system, and it can be collected at the end of the rotor in a pure form. But since the partition coefficients of the two compounds are really close, the two compounds cannot be resolved in just one run, and that is the point when we can switch the role of the phases and the pumping direction of the phases, and just shaved compound E. It means collect compound E and switch to descending mode. It means that we can, in this case, pump the lower phase and collect at the end of the run the pure form of compound B. So once it has been done, it can be done in multiple cycles. So in this way, ascending, descending, and ascending mode, here you can see that the last step, compound E, can be collected in pure form. So just repeating these cycles, the two components can be resolved and collected in pure form. But I forget to mention that when we switch between descending and descending modes, the reciprocal value of the KD values should be considered. It means that if in ascending mode, compound E elutes as the first one, if we change to descending mode, that the compound E will be retained more in the system. Okay, and coming back to the Remdesivir story, here you can see a pilot scale CPC run performed in this multiple dual mode operation with this quaternary solvent system on a dual rotor RCPC setup. Here you can see the running parameters, but I wanted to emphasize that it contained 1.5 MDM cycles, so running in descending, ascending, and descending mode. It means that we started the elution in descending mode and reverting to ascending mode and shaving the compound, the SP diastereomer, and pushing back the mixture of the two compounds and recover in descending mode the two compounds. And here you can see that we have achieved a really nice resolution between the two diastereomers. Okay, and as I mentioned, there is also another option to increase the resolution in CPC by using dual rotor systems. It means that if two CPC rotors serially connect, if we use two rotors serially connected and introduce the feed between the two rotors, then this cyclic process, meaning that switching between the two ascending and descending modes and repeating it as a multi-cycle process, even really challenging separation task can be managed in this way. And what is more important, that in such way the chromatographic separation can be managed in a continuous way, meaning that the injection can be performed in continuously or semi-continuously. Okay, here in this chart you can see the RotaChrom setup of this dual rotor system containing two rotors working in the same mode, a lot of valves, two pumps, one for the feed, one for the eluents, and the same approach. Here you can see working the two rotors in descending modes and pumping, meaning that we pump the lower phase, the more dense phase as the mobile phase, and injecting the feed between the two rotors and collecting product A on the left-hand side. And if we're switching to ascending mode, meaning that it means that we're pumping the upper phase to the system from the left-hand side and injecting the sample from the, it's really important injecting the sample in the upper phase, just not disturbing the hydrodynamic equilibrium of the system. We can collect in this way the compound B on the right-hand side of the system. So this technique has a lot of naming in the literature, such as true moving bed, sequential CPC, or trapping multiple dual mode operations, but it stands for these dual rotor approaches. Okay, and I guess the most interesting and most significant result of this Remdesivir story is the trapping MDM separation of the two diastereomeres. Here in this chromatogram, you can see that the two diastereomers cannot be resolved in a simple batch experiment performed in descending mode, so the overlap is really significant. But once we use this multiple dual mode in a dual rotor system performing 20 cycle, then in this case the rare RP isomer can be isolated in a really high at a really high purity, higher than 99%, as can be observed on the right-hand side in the NMR and HPLC results, representing the crude and the isolated RP isomer in a really high purity. So in this way, we were able to isolate the RP isomer, even starting with a not an optimal feed containing the RP isomer as the less abundant compound. Okay, as a summary of our results, so we tested a lot of scenarios, a lot of inputs, and a lot of modes, so to say. So simple batch solution, multiple dual mode, and dual rotor systems. In this table, you can see two devices, this lab scale dual rotor system and this pilot scale dual rotor system of RotaChrom. In the first line, simple batch solution data is represented with a nice purity but a low yield values on a lab scale system. Here you can see the presented trapping multiple dual mode results of the targeting the RP isomer with a really nice purity, moderate yield, and moderate productivity. And last but not least, the isolating both isomers from a mixture on a pilot scale is represented at the moderate purity level 95%, nice yield values, and what is more important, with increased productivity values. So in this table, I'm pretty sure that it's really simple to realize that this approach is efficient and scalable. Okay, coming to the conclusions of this story, some chemistry related story. Coming back to the sulfobutylator beta-cyclodextrin remdesivir complex. So yes, there is a stable complex reported in the literature used in the infusion of the compound with this inclusion complex described by NMR studies, but in our experiments we were unable to identify selectivity using significant selectivity using cyclodextrin. So our conclusion is that there is no 3-point interaction enabling the chiral recognition of the two diastereomers by cyclodextrin. So most probably the proximity of the chiral phosphorus center and the sugar moiety is not sufficient to get this chiral selectivity. The most important and most significant achievement during the solvent system screening is that we identified methyl THF as a co-solvent in alkanes that enables good solubility and good selectivity for this diastereomeres. And in my perspective, is also an important point to please give a chance to non-chiral approaches in drug-like diastereomer separations, as can be observed in this chart that the non-chiral selectivity values in reverse phase conditions or in CPC conditions that we developed can match the chiral approaches in this case, or even exceed for example, the capillary electrophoresis approaches or HPLC approaches. So please consider the non-chiral approaches in diastereomer separations. And the take-home message of this webinar is that such challenging separations can be managed by advanced operation modes of CPC, even in continuous mode, and didn't represent these results, but believe me, it's also possible to use this approach in continuous mode. And I'm really proud to present here our dual rotor system coming the very beginning of next year with these pilot scale CPC devices, the pumping and valve setup and the control unit on this picture. And I'm also really proud to share that the presented methodology is now a pending patent application. And finally, I have to acknowledge our partners in this study, the Cyclolab, the Hungarian-based cyclodextrin company, the Technical University of Munich, helping us in the optimization of multiple dual mode methodologies, and the Semmelweis University for NMR, providing NMR access and NMR measurements. And with that, I would like to thank your kind attention, and I'm happy to take questions.
Thank you very much, Arpad, and congratulations on this very extensive research and study that you just presented. And as you mentioned, I do indeed would like to ask you a couple of questions. I have three that I want to ask you, so let's go one by one. Firstly, in the realm of chromatography, there is an ongoing debate about batch versus continuous operation. And I'd like to ask you if you could explain the factors that make it viable to switch to continuous operation, and what drives this decision in the context of chromatography.
Okay, yes, this is a super relevant topic, I would say. So in the first approach, it must be clarified that not all the downstream processes can be, or so to say, versed to be implemented in continuous mode. It means that typically an input material containing 2 or 3 compounds, so binary or ternary input material, is needed with sufficient resolution, sufficient chromatographic resolution to be able to adapt this kind of approach. And I have to also emphasize that a high throughput is also a must in such a separation task. And yes, to develop a continuous, for example, a dual rotor methodology, a careful optimization is needed. But the key word in these cases is the intensification. So in these cases, the continuous operation mode provides much higher efficiency at the same solvent consumption, and so resulting in much higher productivity. So the main point in this comparison is that if we considering this dual rotor setup of CPC, I'm pretty sure that this is an exceptionally good fit for such purposes. And last but not least, please let me add that the CPC has an intrinsic ability, since just using liquids in a flow, that it has a ability to be coupled to flow chemistry. So prior CPC and post CPC, also some formulation processes or formulation steps can be also coupled to CPC separations.
Okay, perfect. Makes a lot of sense. My second question would be to touch on the use of cyclodextrins in solvent systems, which seems to be a topic of financial concern for several people. My question is the utilization of cyclodextrins in solvent systems cost effective? And could you please elaborate on the justification for this choice from a practical standpoint?
Yeah, absolutely. So this is a very good question. Preparative separations are always governed by the economics, so to say. And yes, I have to admit that using cyclodextrins in solvent systems, especially at larger scales, cannot be considered as feasible in the first approach. But once we are considering that the target compound can be collected at the end of the CPC separations in the aqueous phase, containing the expensive cyclodextrins. So in considering this case, it can be feasible, and for example, utilizing these cyclodextrin complexes of the target compound in the next formulation step. And another point here that we are working to find efficient recycling options for cyclodextrin containing solvent systems.
Okay, thank you for addressing that. Makes a lot of sense. Finally, I would like to ask one more thing. We've seen promising results with CPC for diastereomeres, but what about enantiomers? Can CPC effectively separate enantiomers as well?
Yes. The answer is yes. There is an option to realize chiral recognition also in liquid-liquid state separations, and there are a lot of successful chiral separations performed in CPC in literature, and the number is increasing by year. The trick here, just as in preparative HPLC, to find the chiral selector of the separation and optimize the solvent system accordingly. So let me just mention that cyclodextrins and other chiral selectors has been utilized successfully to separate, for example, different azole APIs with a huge selectivity values, resulting in nice high throughput and well resolved liquid chromatographic methodologies.
Okay, thank you very much, Arpad, and for sharing your expertise and all these exciting pieces of knowledge about chromatography. Once again, congratulations to you and the whole team participating in the research. And with that, I am going to wrap up today's webinar. We hope you found today's discussion exciting and informative. Again, another big thank you for our lead scientific officer, Arpad Konczol, for sharing his valuable insights and expertise with us. And with that said, see you all in the next webinar. Goodbye.
Goodbye.